Good evening, students. I'm Rekha from Prakara. I hope I'm audible to everyone. So, how are you all? So, shall we wait for some students to join in early? Fine, great. So, good evening, nurses. So, from Prakara, I will be dealing with this topic called structure and classification of microbes. Why we need to learn about structure and classification of nurse, being a nurse? Why should we learn about microbes and all? Yes, we need to learn about microbes because during the COVID era, you equally fought with doctors against this COVID virus. So it is necessary for you people to learn about these microbes. Unless and until if you know the basics about microbiology, you can't secure patient from getting reinfected to protect them from hospital acquired infections and from different pandemics. As a nurse, you need to support them throughout your entire life. You need to save different patients from different infections. So it is necessary for you people to have a basic idea about microbes. So today, now in this class, I will be giving an introduction about morphological types, size and form of bacteria, motility, colonization, growth and nutrition of microbes, temperature and moisture. So I'll be dealing introduction about these topics. So as a nurse, you should have a command about these topics. So why you need to have a command? So unless and until you know about stuff or yes, then only you can save that person, like when to give right medicine, right dose at right time. Only a nurse will be staying along with the patient to have proper antibiotic at appropriate time. If the dosage time is missing, yes, if some other antibiotic was given to that patient, he might develop resistance to that antibiotic. In other time, when he got admitted, when the same antibiotic was given, high dosage need to be prescribed for him. So to save the patient from reinfection and also to protect him from hospital acquired infection, you need to learn the basics about microbes. So, you might be learning about the same content, structure and classification from your class, from your books, Google, internet, YouTube. So, everywhere you will be learning. So, but in Prakara, we are helping you to cover up those loopholes wherein which you are missing as a nurse. So, if you want to become an ICN nurse, like what are the gist and what are the basic techniques that you need to learn. So everyone can become a nurse. But to be a proper dedicated nurse, you need to learn about few dedicated techniques, which we are dealing at Prakara. So we want you to come as an flying colors. That's the reason why we are supporting you. So coming to the topic proper, coming to the structure and classification of microbes. So what is this? uh, Structure, coming to the structure and classification of microbes. So in 17th century, this a great scientist called Linnaeus, he established this taxonomic rankings as domain, kingdom, phylum, class, order, family, genus, and species. Why we need to learn? So as a human, it is necessary for us to learn about our family history. Who is our father, father's father, fourth father. When we are learning about our family history as a human, so how our animalia, our domain has exist. So when we are naming, we'll be telling our family name with daughter name. Say for example, what is our surname? We'll be naming with our surname with our name. So in the same pattern, as a microbiology people, you should, as a nurse, you should able to learn how does that microorganism has exist. So usually these organisms are classified as first as domain. So the domain includes eukaryotes. There are two different domains like eukaryotes and prokaryotes. Like single celled, without there is a appropriate nucleus is there with particular nuclear material, it is that falls under eukaryotes. If inappropriate nucleus is present without a nuclear lining, they are called as prokaryotes. So and all the animals which are able to move, They falls under kingdom Animalia. Again, this domain was divided into three kingdoms: Animalia, so plantae, different. So kingdom Animalia includes the organisms which are able to move, and the phylum Chordates. So phylum Chordates, those are the organisms which has backbone like us, like our spinal cord supports as a backbone to us. Coming to class Mammalia, so those organisms which has hair or fur and which has milk glands, they falls under class Mammalia. Coming to order primates, why they were called as primates? Those who are having collar bones, which you have already learned in anatomy subject about the collar bones. These are the collar bones. So they falls under the order primates. Coming to family hominids. So those organisms which has flat faces and three-dimensional eye view falls under hominidae. And genus Homo, that is organisms which has upright posture, so, and large brains like humans falls under Homo. Species like Homo sapiens. So, those organisms with forehead and thin skull bones falls under Homo sapiens. This is the main taxonomic rank. Say for example, now we are dealing with bacteria, that is microbes. So the main domain is bacteria. It belongs to phylum Proteobacteriae and class like alpha, beta, gamma, and delta. Order, again, there is different orders like Enterobacteria, Pseudomonas. I'm just giving an example about a bacteria. This is how each domain was differentiated depending on the features. Like family, Enterobacteriaceae family, it has different microbes like all gram-negative. All hospital-acquired infections are caused by these bacteria like E. coli, Proteus, Pseudomonas. These are the different bacteria which causes hospital-acquired species. And they are further divided into different species. Say, for example, Shigella. Shigella boydi, Shigella dysentery, Shigella flexneri. These are the different species. If you take E. coli, again they are divided into Escherichia coli, like that. Each is divided. Next, the great scientist Carl Linnaeus was the person who has introduced binomial nomenclature. Just like our humans, we were named with our surname. For example, Koppala Rekha. Koppala was surname. Rekha was the species. In the same pattern, each organism should have two names, one genus and one species name. Like, for example, Homo sapiens. So he was the great person who has introduced this binomial nomenclature to introduce different microorganisms. Say if you go in bacteriology, Escherichia coli, Shigella dysentery. Shigella was the genus and dysentery is the species. So each bacteria will be named with their genus name and species name. Next, later in 19th century, Carl Woos was the person who was divided the domains into three domain systems like archaea, bacteria, and eukarya. So this is the phylogenetic tree which represent bacteria, archaea, and eukaryote. So now we will be learning like how each individual was different from different things. So archaea was the prokaryote which has undefined cell wall. And eubacteria or bacteria are single cell prokaryotes and they don't have well-defined nucleates. And eukaryotes are the organisms which are multicellular organisms and they have defined nuclei. So depending on the nucleus, they are divided into bacteria, archaea, and eukaryotes. So later in 1937, Charton was the person who has divided these biological groups based on the cellular organization. Like earlier was the person, he has divided into archaea, eubacteria, and eukarya. Later, Charton was reintroduced the same thing with indefined manner. Like for example, prokaryotes, as I have explained, they lack well-defined nucleus. Whereas eukaryotes, they have appropriate nuclei with organelles like ribosomes, mitochondria. These are the cell organelles. They have defined nuclei. Like eukaryotes includes plants, animals, we humans falls under this. Prokaryotes are like bacteria. They don't have well-defined nucleus. So depending on cellular organization, their morphology, nutrition, these bacteria are classified into different groups. For example, one such group is algae. Algae, what you remember, you might have seen these algae in rivers, lakes, ponds. In Telugu, we might say pachi. So when there is a stagnant water in bowl, you can see this algae. What are these algae? So they can be either unicellular or multicellular, and they have well-defined cell walls. As it is green, you know it is photosynthetic. That is autotrophs. It can synthesize its own food. So where we can see usually in rivers, lakes, ponds, we can see this. So usually it will reproduce by two modes, sexual reproduction and asexual reproduction. So there are few asexual methods like binary fissions, fragmentations, or conidia formation, which we see for different organisms in different pattern. For algae, usually it will reproduce by cell division. If it is sexual mode of reproduction, it is by fusion of gametes. It occurs. So example for this algae are chlamydomonas. Next is archaea. Archaea are unicellular prokaryotic organisms and it also has well-defined cell wall. They are also autotrophs. Like they can be either chemoautotroph or photoautotroph. That means they can utilize the light to synthesize its food, else it utilizes chemicals to synthesize its foods. Usually these archaea microorganisms will be seen in extreme environments like hot springs, highly saline environment, acidic habitats. Like in deserts, there is oasis. There also you can see this archaea. Else in foreign countries, usually in US ponds, lakes, you can see because of this extreme or highly saline content in oceans also you can see this archaea. And they also reproduce asexually by binary fission and sexually by gene transfer. Gene transfer can be done by three modes: conjugation, transformation, and transduction. So example for this archaeobacteria are halobacterium and pyrococcus. Next, coming to our topic of interest is bacteria. This is also a group of bacteria. These are single cell prokaryotic organisms. So again, these bacteria are divided into two types depending on the presence or absence of this peptidoglycan layer. This archaeobacteria don't have peptidoglycan layer. Here you can see only cell wall is there and defined nucleus is there. Whereas in case of this algae, you can see this multicellular, unicellular with defined cell wall, but there is no nucleus. Here, nucleus is there, cell wall is there, but peptidoglycan is not there. But in case of bacteria, there is a designed peptidoglycan layer, which is absent in other two. Okay, these are ubiquitous and they are present in different habits. Like they might be, we can see bacteria in soil, air. As a bacteria, it might present in our skin also as a flora of our body. So usually their mode of nutrition is autotrophs or heterotrophs. They either can synthesize their own food, else they depend on organic products to synthesize their foods. They usually reproduce by binary fission, budding, spore formation, and sexually is through transformation, transduction, conjugation. These are the three modes of sexual reproduction. So examples of bacteria are plenty like E. coli, Salmonella, Pseudomonas, Proteus, different Streptococcus, Pseudomonas, which you will be learning in systemic bacteriology. Next is fungi. What is this fungi? Have you come across with some fungi which is appearing on bread or groundnuts? If you have stale food, there you can see some spiders or some fungi. So that is nothing but fungi. Those are molds, we can say. If it is single cellular, we call it as, in our terms, we call it as yeast. If it is multicellular, we will call it as mold. So you keep groundnut, dip it in water and keep it as such for 3-4 days. You can see the development of mold. You keep the bread and leave it for a while for 3 days. You can see the development of mold. That is nothing but fungi. If you send it for microbiology, if you do the T's mount, you can easily able to appreciate Aspergillus niger, which is responsible for food poisoning. So now this fungi is also multicellular. So whatever we are seeing is multicellular. That is nothing but mold in microbiological terms, we call it as. So they are also eukaryotic microorganisms. They have well-defined cell wall and other cell organelles. So next, other group of microorganisms which are classified into other group is protozoa. So they are single cell eukaryotic microorganisms like entamoeba histolytica, food poisoning we can say. If you are like, you might be having like ordered some Swiggy, like you might be having food poisoning. You have a complaint of stomach ache. You will be approaching doctor, and doctor you can say, sir, at the right hand side I have a stomach ache and I have uh, motions for three to five times. So what does it indicate? Food poisoning with protozoans, nothing but entamoeba histolytica. So it is an eukaryotic microorganisms. It has complex internal structure. They are motile. That's how it is able to create the pathogenesis, and it is able to damage our intestine too. So they reproduce sexually by isogamy. It's a parasite. Amoeba, entamoeba, histolytica, plasmodium, all parasites falls under these protozoa groups. So these protozoas are also eukaryotic microorganisms, and they are motile too. They can present, as you already know, food poisoning occurs through water and through food. So which indicates it can inhabit it in soil and water. Next are slime molds. So these slime molds are either single cell or acellular. That means it can contain either single cell or without cell. It forms a slime-like layer on the host organisms. So their mode of nutrition is usually holozoic. Holozoic I'll be dealing with in next slide. So usually they will reproduce by forming spores. So example for slime molds forming microorganisms are Dictyostelium. Next is viruses. This you should learn since you have dealt with, will be dealing with many pandemic cases in future. So it is necessary for you to learn about viruses. What are these viruses? They are biological entities, and they won't replicate by either by sexual or asexual method. It will replicate through DNA. They either will replicate along with host DNA, else they will introduce their DNA into host DNA and damage our structures. That is how coronavirus has worked. That is how its pathogenesis has been occurred into our body. So based on that type of genetic material, whether it contains DNA and RNA, they are classified into DNA viruses and RNA viruses, depending on the genetic material. The DNA strand is single strand, they are called as single strand DNA virus. If the DNA has two strands, then it is called as double stranded DNA. RNA again is the same, single stranded RNA or double stranded RNA. Depending on the strands and genetic material, these viruses are classified into different groups. And so this nuclear material, that is DNA or RNA, is enveloped by a capsid. That depending on the capsid shapes, again these viruses are classified into different types. Spiral capsid, polyhedral capsid, spherical capsid, or complex structure capsids. So as I told you, usually they can't replicate outside the environment. They can able to replicate into suitable host. It is replication is done by two cycles, that is lytic cycles and lysogenic cycles. So lytic cycle is nothing but the virus genetic material will develop into our body. Later it will cause infection. That is lytic cycle. In case of lysogenic cycles, it will incorporate virus DNA into our DNA. Then it start replication. Then each and every part of our body will contain this virus, which will be transferred to different hosts. That is how the spread of infections will occur. As the virus DNA is also incorporated into our DNA, when our process is going on in our body, along with our steps, this virus DNA is also started replicating. That is how it grows. That is the reason why we were not able to stop even COVID virus, that is coronavirus, or HIV virus, or hepatitis B virus. There is no prompt treatment for HIV. There is no cure. But we can prevent the patient from getting reinfections from different bacteria or different viral infections. We can save that. We can extend the lifespan of the patient in case of HIV or COVID or in hepatitis B infection cases. The reason why, because the whole virus DNA is incorporated into our DNA. That's the reason why there is no dead-end treatment for these viral infections. So next is the classification depending on the gram staining reaction under cell wall. As I told you, like bacteria are different from RK and the like eukaryotes by the presence of cell wall called peptidoglycan, which is very much important in staining technique and also to differentiate the gram positive and gram negative organisms. If it has thick peptidoglycan layer, what do we call it as? They are called as gram positive. So they are basically divided into three groups. Those bacteria which has rigid cell wall, they are called as gram-positive and gram-negative bacteria. Gram-positives like Staphylococci, Streptococci, gram-negative like E. coli, Proteus, Salmonella, Shigella, and acid-fast bacteria like Mycobacterium and Rickettsia. These all falls under the bacteria which has rigid cell wall. And some bacteria will have thin or flexible cell wall. They falls under Treponema. And those bacteria which lack cell wall, they falls under mycoplasma. Okay. Say for example, if we have cold, if we have thick sweaters, they falls under rigid cell wall group. That is gram positive, gram negative. That is how they are were able to resist when they enter into our stomach. So when we ingest either some bacteria through fecal-oral route, our host enzymes were not able to digest the bacteria. Why? Because it has the thick shield, thick peptidoglycan layer. That is a protective layer for it. Okay, some bacteria might be having thin flexible layer. Those are called spirochetes. Those bacteria which are lacking cell wall, those are called as mycoplasma. They falls under that group. So these are the few examples for gram-positive, as I told, staph, strep, clostridium. And gram-negative bacteria, as I told you, E. coli, Serratia, Vibrio, all these falls under gram-negative. Next is the classification depending on peptidoglycan layer. That is gram-positive, gram-negative, thick peptidoglycan, thin peptidoglycan, and those bacteria which lacks cell wall. So next is the classification of bacteria on the basis of oxygen requirement. Like they are classified into aerobes and anaerobes usually. So aerobes are the bacteria which grows in the presence of 21% oxygen. They are called as aerobic bacteria. So anaerobic bacteria are the one which can survive in the absence of oxygen. They are called as anaerobic bacteria. Next is facultative anaerobic bacteria. They might require, they can grow with the presence or in the absence of oxygen. They are called as facultative. Oxygen is there or not, they can survive. Microaerophilic, if little quantity of oxygen is there, that is 2 to 10% oxygen is there, those bacteria can survive. See, if your lecture is guiding, so the students will study only if the lecture is always pushing them. They are called as aerobic people. So when there is no lecturer, then they can't feel stress. They can read properly and can write properly in exams. They are called anaerobic bacteria. Either in the presence of staff or the absence of staff, they can survive betterly. They can do exams betterly. They are called as facultative. On and off, if the staff is coming and going, the people able to study, student able to study. Those are falls under microaerophilic bacteria. Absence, presence, some they might present or absent. They are called facultative. If their presence may not do any difference, they are called as microaerophilic bacteria. Next, depending on the pH requirement, they are again classified into 3 types. So what is the normal pH range? 0 to 14 is the normal range. 0 to 7 is considered as acidic. So 7 is neutral. Again 8 to 14 is considered as alkaline. So depending on the growth of these microorganisms at that particular pH range, they are classified into 3 types like acidophiles, neutrophiles, alkalophiles. Acidophiles are the bacteria which grow at 0 to 5. Neutrophiles, as the name itself indicates, neutral pH. They can grow between 5 to 8. Alkalophiles, so alkaline pH. So as the name itself indicates, they can grow beyond 7, that is 8 to 11. So again on the basis of the salt. So these on depending on their growth, nutrition, size, structure, depending on these characters, they are classified into these groups, which I am explaining. So again on the salt intake and the resistance to the salt, they are again classified into 2 types. So this, when we are learning about salt, depending on the salt requirement, we can able to categorize the bacteria also. So if the bacteria was can't able to tolerate in salt condition, we can apply that salt in antibiotics. Else we can see, we will be doing homeopathy. So there also we can apply this salt to reduce the infection. So that's the reason why we it is necessary for us to learn about even this minor topic. As a nurse, you should be aware of this. Then only you can help the patient to get for better recovery from the infection. So based on the salt requirement, they are classified into two groups, like halophilic or non-halophilic, which can survive in the presence of salt. They are called halophilic bacteria, which cannot survive in the presence of salt. They are called non-halophilic bacteria. So that's the reason why when we have throat infection, people asked us to do goggling. So those bacteria cannot survive in saline condition. For example, streptococcal infection, if you have throat infection. So doctor will advise us to do salt water goggling. Why? If you learn these only, you can read that. You can advise the patient to do, because it can't survive during salt condition, in saline condition. It can't grow. So that's the reason why we need to learn about this classification to have better understanding. It is not necessary just to learn about, okay, this is E. coli, this is staphylococcus, okay, this is the morphology. It's of no use. If you learn about these basics only, it will be helpful for you to treat patient. So as I told you, so they are halophilic and non-halophilic. Again, halophiles are divided into four types. That is halotolerant, slight halophiles, moderate halophiles, extreme halophiles. Like depending on the NaCl concentration, salt is nothing but NaCl. Depending on NaCl concentration, like if the bacteria able to survive to 5 to 10% NaCl, they are called as halotolerant. If it is able to survive 1 to 5%, it is called slight halophiles. If it is able to tolerate 5 to 20%, they are called moderate. If they can able to tolerate between 20 to 30, then they are called extreme halophiles. So examples for the same. Halobacteria like staph, slight is halomonas, and moderate is bacillus, and extreme is halococcus. You need to learn about these examples to treat patient well. So when we'll be learning about systemic bacteriology, at that time you will be learning in detail about each organism. There you will be having better idea about each organisms. Again, depending on nutritional requirement, as we are taking different sources like carbohydrate source, protein source, and nucleic acid source, like the same, the bacteria also require, the microorganisms also require sources like carbon source, energy source, and electron source for its survival. How it is making. If we learn about their source, their metabolism only, we can give define particular antibiotic towards it. Like if we know what is the carbon source for it, we can restrict it. So in earlier days, people were put in unmanned jail. So they were given with lesser amount food, because to restrict output. So that is how they used to ill treat. It's the same pattern. If we know the carbon source, energy source of the bacteria, we can restrict the growth pattern of bacteria by knowing its sources of nutrition. So there are three main sources for this bacteria, or these microorganisms, to prepare food. One is carbon source, another is energy source, and is electron source. By making use of three sources only, it was able to get nutrition from host, that is nothing but humans or animals or plants. So depending on carbon source, they are divided into two types, autotrophs and heterotrophs. Autotrophs are the bacteria or the microorganism which can, autotroph as the name itself indicate, they can self-synthesize their own food. You have read about photosynthesis in your 10th class, right? What is the principle? So in presence of sunlight, by utilizing CO2, it will convert this into energy by making utilization of water, H2O, as an electron source there in the same pattern. So what is the carbon source here? CO2 is the carbon source. So if it makes its own food, they are called as autotrophs. So if it gets that food source from organic compounds, that is depending on other compounds, they are called as heterotrophs. Okay. So if some cat kill mouse, if it get foods, they are nothing but heterotrophs. They are killing some organism and getting nutrition. They are called heterotrophs. So plants, they can synthesize their own foods. They are called as autotrophs. So it is the same. So if it synthesize their own food, they are called autotrophic microorganism. If they depend on other sources, they are called as heterotrophic organisms. And next is source of energy. The earlier one is depending on source of carbon. Second one is source of energy. So if it utilize, like if you remember photosynthesis principle, so there also it will utilize sunlight, carbon source, and water. So carbon source we have told. Next is light source. So if it utilize light as a source of energy, they are called as phototrophs. If it utilize oxidation of organic and inorganic compounds, then that if it utilize that sort of energy, they also falls under chemotrophs. So chemotrophs are the organisms which derive energy from by oxidation of organic and inorganic compounds. They are called as chemotropes. That is second classification. Third classification is source of electrons. Next, to synthesize food, three modes. That is, I have told you about carbon source, energy source. Third one is electron source. So if they utilize electrons from inorganic compounds, they are called as lithotrophs. Else, if they utilize electrons from organic compounds, they are called as organotrophs. As the name itself indicates, if they utilize energy from organics, they are called organotrophs. If they utilize energy from inorganic compounds, they are called lithotrophs. Okay. So next classification of bacteria is depending on the shape and size. As the name itself indicates, if they are circular or spherical, they are called as cocci. If they are in rod shape, they are called as bacilli. If they are snake shape, like spiral shape, they are called as spirilla. If they are comma shape, as the name itself indicates, comma shape bacteria, they are called as vibrio. And next is so there are, depending on the presence of flagella, they were again classified into two groups, that is aflagellated and flagellated bacteria. So this will be dealing in further. So that depending on the ability to form spores, they are again further classified into two types, that is spore formers and non-spore formers. Spore formers can be Clostridium, Bacillus. Non-spore formers like E. coli and Staph aureus. And again, spore formers are classified into two types, exospores and endospores. So depending on spore formation, they are classified into spore forming, non-spore forming. Non-spore forming gone. When we are studying spore forming, again, if the spore is formed outside the cell, then it is called exospore. If the spore is formed inside the cell, endo means inside. Inside the cell, it is called endospores. Next, capsule. These are the secretions caused by the secreted by the bacteria to protect from host as a host defense mechanism. So if they produce capsule, they are called capsulated bacteria. And if they don't have capsule, they are called as non-capsulated bacteria. This capsule plays a key role in the like admitted patients. Usually, Klebsiella, they form capsules. Once that matrix formation occurs, it will attract all the bacteria in the hospitals, and there will be formation of biofilm, which is hectic for the doctors to treat antibiotics to get the patient to get rid of those infections, because multiple bacterial infections will be there because of this capsule. Once the organism has capsule, then it is started producing slime, then it will form that matrix. It will attract all the bacteria, all the viruses. So when we are sending that sample for microbiology lab to do the culture, you will able to recognize different bacteria. It is not one or two bacteria. If it is one or two bacteria, we can give two antibiotics. So plenty of bacteria will be there. So for the clinician to treat that patient also, it is very difficult because he was supposed to take different types of enormous antibiotics. So that is the reason why you need to learn about capsulated bacteria and non-capsulated bacteria. So this was one frequently asked question in your previous examinations. Write about the classification of different microorganisms with examples. So this is next group, next classification. Depending on the respiration. So they were again classified into two groups, aerobes and anaerobes. Okay, this is general classification. So subgroups among it is aerobes. So what is meant by aerobes? They can survive only in oxygen, the presence of oxygen. Anaerobe means no oxygen. The bacteria can survive in the absence of oxygen are called as anaerobes. Obligate aerobes. That means they should have proper oxygen supplement is the mandatory thing. If oxygen aerobes or they can survive in whatever the concentration of oxygen. Obligate aerobes are the organisms. They should have 20% oxygen. That is, oxygen is mandatory for those organisms to survive. So anaerobes are the bacteria which can survive in the absence of oxygen. And next is facultative anaerobes. So they can grow with or without oxygen. They are called as facultative anaerobes. Obligate anaerobes. So they will not survive. Obligate means mandatory thing. They should require. Obligate aerobes means they should have oxygen. Obligate anaerobes means they should not have oxygen. Okay. Mesophile, as the name itself indicates, they should have moderate oxygen levels. Next, this is the same classification depending on the coccus arrangement. So in the structure shapes, we have learned about two shapes. What are they? Cocci and bacilli. That is spherical shape and rod shape. So again, the arrangement of, depending on the arrangement of cocci, they are classified into different types. If there is single cocci, it is called a single coccus. If there is two cocci, it is called as diplococci. So you can see in pneumococcal infection, like lanceolate needle shape, you can appreciate through microscopy. So example for diplococci are streptococcus. Example for single cocci are staphylococcus. Next is tetrads. So if the cocci are arranged in four, they are called as tetrads. Next is if the cocci is arranged in chain-like fashion, those are called streptococcus. If the cocci is arranged in grapes, grape-like structure, like triangle form, which you can see. So those are nothing but staphylococci. If they are arranged in cube-like cocci, they are called as sarcina. Okay, single cocci, diplococci, tetrad, chains, streptococci, grapes like cluster, they are called staphylococci. If it is in tetrads, it is called sarcina. So in the same pattern like the cocci, bacilli also depending on the arrangement of bacilli, they are classified into single bacilli, that is one single rod. Diplococci, that is diplobacilli, that means two rods together. Streptobacilli, same chain of rods, like you see in bacillus. Diplococci, you can see in Coxiella. Palisades. So side by sides, the bacterium will arrange. That is called palisade manner. Next is Coccobacillus. That is oval. It will be rod only, but it is in oval shape. Like Haemophilus influenza, you can see. Vibrio, like I have shown in shapes. So comma-shaped bacteria. That is clear. That is a significant feature for Vibrio. Rice water stool, you can see in case of Vibrio. So there was an era people used to suffer a lot about a Vibrio that we'll be dealing in systemic. I'll tell you a story later in while dealing with Vibrio. So next is spiral. Spiral shaped bacteria are called as spirilla. Next is spirochetes. That is corkscrew bacteria falls under spirochetes. So other shapes like filament shape. Filament shape is nothing but a slight thread. Next is club. Club is nothing but gather, which use like Yama Dharmaraja used to be holding, no? The same, it's club shape, whatever he is holding. So that is in club shape. So Cornybacterium usually shows this club shape feature during gram staining. Next is star shape. So stellas will be showing in star shape. Next is pleomorphic, like Mooservelli used to say in Telugu. Like they used to change, they don't have particular shape only. Those bacteria are called as pleomorphic. So these bacteria, usually their size will be represented in, like if you length, we will represent in meters. In the same matter, in the same form, bacteria also will be bacteria, viruses, fungi, parasites will be represented in uh, uh, particular measurement dimensions. They were represented in angstroms, nanometers, micrometers, and millimeters. These are the units in which we used to represent the size of bacteria, viruses, and parasites. So usually bacteria will have 0.5 to 2 micrometer size. It might be less or more than that. Okay. So this is a picture showing relative size of different bacteria, viruses, and parasites. So next, coming to properties of bacteria. One particular property is motility. So what is motility? The ability to move. So motile, we are motile. What does it mean? Motile means movement. So the ability of a bacteria to move is called as motility. So few bacteria motility will be achieved by three factors: flagella, fimbriae, and pili. If this bacteria possess either of these property, they can able to move. So first thing, as I have mentioned, flagella. This is the structure of flagella. This is locomotory organ in bacteria. So flagella has three parts like filament, hook, and basal body. This filament is attached to hook. It is freely moved, and because of this filament, the bacteria are able to do swimming motility. So propulsion, forward and backward movement of this filament, flushing of this filament, the bacteria are able to move. This filament is made up of flagellin protein. So this hook holds basal body. So depending on the presence of flagella, again they are classified into different types. Atrichous. If the bacteria has no flagella, they are called as atrichous. If the bacteria has single flagella, they are called as monotrichous. If the bacteria has single flagella on both poles, they are called amphitrichous. If the tuft of flagella are present at one side, they are called as lophotrichous. I am showing it in image. So if the tuft of flagella is present at both the ends, it is called as amphitrichous. If the flagella is present entire body, they are called as peritrichous flagella. You need to learn about one example for each, like spirochete for atrichous, Vibrio cholera for mono, aquaspirillum for amphitrichous, E. coli for lophotrichous, Campylobacter for amphilotrichous, and Salmonella for peritrichous. So what is the function of this flagella? It is helpful for motility, and it is also helpful for signal transduction, and it also acts as a virulence factor. It will also helpful for the attachment of bacteria to the host cell. So next motile fragment is pili. So these are short hair-like tube structures which helps in attachment and also for reproduction it will help. It is made up of pilin protein. So again, it is of two types, long conjugated pili and short pili, depending on the function. So long conjugated pili helps in conjugation and short pili helps in attachment. So again, depending on the structure, they are classified into four types, that is transfer pili, type 4 pili, type 1 fimbriae and curly fimbriae. So coming to fimbriae, this is also one of the locomotory organ. So flagella, we have read the structure. So pili, they are hair-like structures. So these fimbriae are also small fiber-like structures. They are present on the bacteria. This also helps in attachment and also motility. Okay, so now we are talking about motility. All bacteria are not same. Our walking styles are also different. Like Rajnikant has different walk style. We have different walk style. Men will walk differently in the same pattern. All the bacteria will have different walking styles. So motility styles. So that depends on number of flagella. If the flagella are more, they will have different motility. If there is less flagella, they will have different motility. And if they have pili, the motility will be different. Okay, why we are talking about this motility? So it is helpful for identification of bacteria. If we learn about motility, for example, protease, if you take, protease will exhibit swarming motility. By seeing the growth of the protease itself, we can identify looking at the agar plate. When we send the patient sample, we will do the streaking, right? That we will be learning in culture. What do we do with the patient sample? So once the streaking was done, the entire bacteria will spread in the entire agar plate. So that's because of motility. This motility, if you learn about motility, it is helpful for identification. So if it will take nearly around some 3 days to identify the bacteria and to prescribe to the antibiogram, like which antibiotic will work better for this bacteria. To learn about that, it will take 3 days. If you know about the motility and if you are able to identify the swarming motility in 1 day, you can treat the patient in 1 day itself instead of taking 3 days. The patient will recover faster. That is the necessary why we need to learn about motility character. Okay, first type of motility is swimming. So as I told you, flagella helps in swimming motility. Next is swarming motility, like that I have explained you. Like protease will show swarming motility. It swarms, that is, it spreads towards the entire plate. That is nothing but swarming motility. Next is twitching motility. It will move like this, like fast, quick and jerky movement called as twitching motility. It will move like this. Gliding means it will slowly glide on the wall. Okay, like some lizard. If the look at the lizard, if it finds some ants, then it will show quick and jerky movement. If it is moving slowly, then it will show gliding movement. On the wall, if you see lizard at your home, if it moves slowly, that is called as gliding movement. If it sees some insects or ants, it will show quick and jerky movement. That is nothing but twitching motility. Okay, these are the different types of motility patterns. Next is slime forming microbes that we'll be learning about biofilm protection. So again, there is an important property called taxis, that is stimulus or attraction towards the certain materials. Like if the bacteria gets attracts towards chemical stimulus, they are called chemotaxis. If the bacteria get stimulated by oxygen, they are called as aerotaxis. If they get stimulated by photo or light, they are called phototaxis. If they get stimulated by temperature, they are called thermotaxis. If they get stimulated by magnetic field, they are called magnetotaxis. If they get stimulated by fluid flow, they are called as rheotaxis. If the stimulation is through moisture, they are called hydrotaxis. If the stimulation is through physical contact, then it is called pygmotaxis. Next is demonstration of motility in bacteria. So once, if it is protease, okay, if we can look at the agar plate, we can see this is protease bacteria. It takes us forming motility. There are different types of motility also. That also we need to identify. How we need to identify? It can be demonstrated by 4 different ways. One is hanging drop preparation. This will be, you will be learning in your practicals, hanging drop method. So next is through phase contrast microscopy, dark field microscopy, and we can stain the flagella also, because flagella is also one of the locomotory organism which helps in motility. By staining that flagella, we can appreciate the motility. And other way is electron microscopy. These are the 4 ways. One is hanging drop method. Next is phase contrast, dark field microscopy, and electron microscopy. Through these 4 methods, we can able to appreciate the motility of bacteria. This is also important frequently asked question in your exams. What is motility? Explain the modes of locomotion in bacteria. So now I have explained you earlier. It's the same explanation for this question. What is the motility? Means the capacity to able to move. Movement is nothing but motility. So more, what are the modes of locomotion like flagella, pillae, all these things you need to explain for this question. So we have gone through your whole previously asked question papers and we have found these questions that we are representing here. Next is important properties colonization. Organisms able to move. So if we are able to move, we are able to form groups, gangs, right? In the same manner, if there is similar properties, the organisms will gather at one place, they will colonize. Okay, it's the same. So now we will be learning. So how that colonization will form. So what does it do? First, it will attach to host cell surface. Okay, like when we are a new person, so we want to settle in Maharashtra. So what do we see? We will inquire some few people, few of our friends. We'll know few of our friends. So then we'll go stay there at home and we'll search for some homes, like some new homes were constructed or not. Then we need to inquire about the electricity source and what are the other immune responses, like host immune responses is nothing but what are the problems that we are facing. Okay, then we will shift to that place. It's the same procedure. So organism will come attached to that host cell. It will obtain nutrients. So it will enter into our body. It will visit to that place. It will sit in their friend's home. We will sit in our friend's home. We will obtain nutrients from our friend's home. In the same manner, organism will take nutrients from our body. It will start replicating from that in our body. So we will shift there. So we will overcoming host immune response. That is the whatever the problems that we are going to face at that house, electricity problem, water problem, those are host immune responses. So organism should overcome our problem, whatever the secretions, enzymes that are antibodies that are targeting on that organism, it should able to resist. So those war syndromes, it should overcome. Then only it can transfer to other host. That is how colonization occurs. So first step is adhesion, that is attachment to the host surface. If the bacteria found some mucus surface, if there is an open wound, you can find mucus. So it can, affiliation can occur there. So it will start binding towards it. Else, if the organism has locomotory organ like villi, it can attach to, because this villi and fimbriae helps, aids in attachment. So it helps, it will easily attach to the host cell. So what does this adhesion will do once it started attaching to host cell? This is particularly important in hospital admitted patients because this adhesion will plays a role in biofilm formation. So the organism will come attack on the stick onto the mucous surface or the operated surface. What does it do? It will form a matrix and it will leave, but it will attract the other bacteria. In that matrix, different bacteria will come and sit on that matrix. It started replicating. So we need to be much more cautious toward that patient. Whatever the antibiotics we might give, So it won't work for other bacteria. The clinician will be treating with one bacteria, but because of this biofilm formation, there will be an engulfment of different bacteria. So the clinician has to give much more antibiotics to treat that patient to prevent that infection. Okay, that's the reason why we need to learn about biofilms. So next is growth and nutrition of microbes. So how it is able to grow, how it is able to get the nutrition. So growth we will describe in the form of size. So nutrition is nothing but how it is obtaining the nutrition, how the bacteria is growing, what are the nutrients it is taking comes under nutrients, how it is obtaining that food that comes under nutrition, how it is growing in size comes under growth. So depending on the nutrition, it has three main requirements as I have told you earlier: carbon source, energy source, and electron source. Coming to the nutrients. So here we have described about nutrients. Two types of elements or nutrients it requires. What are those? Macro elements. Again, this carbon, energy, and electron sources are further subdivided into macronutrients and micronutrients. These carbon, oxygen, hydrogen, phosphorus, these are all called as macro elements, which helps in biosynthesis of biomolecules. Biomolecules you will be learning in biochemistry. Okay, carbohydrates, proteins, nucleic acids, all these are called as biomolecules. So these macronutrients falls under which is helpful for biosynthesis of these biomolecules for the bacteria. And there are certain micronutrients or trace elements like zinc, cobalt, magnesium. These are called as microelements. So what's the roles of these microelements? They act as a catalyst to speed up the reaction. It will speed up the reaction. They act as cofactor. Okay. So when you want to fight with your enemy, it is not necessary you will attack that enemy directly. You will find some other enemy who is enemy to that person. So you will start adding few more things to that person. Then you will speed up the reaction. You will encourage the fighting. It's the same procedure. It won't involve directly. It will encourage its enemy to fasten the reaction. Those are nothing but microelements. So depending on the modes of nutrition, they are classified into different types. As I have explained you earlier, autotrophs, heterotrophs, phototrophs. Autotrophs are the one bacteria which can synthesize its own food. So heterotrophs are the one which get carbon source from already produced sources like organ substances from other organisms. Phototrophs are the bacteria which require light as a source. So photoheterotrophs are the source which require light and organic compounds. They require both. Photoheterotrophs. So photo means light source. They require light source as an energy source and electrons as an organic compounds. Chemotrophs. So they require energy by oxidation of organic and inorganic compounds. Chemotrophs and chemoheterotrophs sounds similar, but the difference is chemotrophs are the one they obtain energy by oxidation of both organic and inorganic compounds. Those are chemotrophs. Chemoautotrophs are the one which get energy by oxidation of inorganic compounds. Those are chemoautotrophs. If by oxidation of if it is by through organic compounds, they are called as chemoheterotrophs. Autotrophs utilize inorganic compounds, but heterotrophs utilize organic compounds. So lithotrites and organotrophs, they're the same. They utilize the organ chemicals and light source. So coming to the modes of uptake of nutrients by microorganisms. So how does the microorganisms take nutrients from it enters into our host body? How it is obtaining like nutrients? It is through different ways like diffusion. First one is diffusion, like passive diffusion. So through passive diffusion, it will obtain O2, H2O. All these things will be obtained through passive diffusion. What is meant by passive diffusion? Movement of molecules from higher concentration to lower concentration. So in the host environment, outside the cell, that is bacterial cell, if there is more oxygen concentration, so oxygen will move from host to bacteria, inside the bacteria. That is how it is surviving. So through passive diffusion, it will get carbon dioxide, oxygen and water. And next one is facilitated diffusion. It is also same. So high concentration to low concentration, it will obtain all the oxygen, that is carbon dioxide and water. But here it will utilize carrier proteins. These are nothing but permeases. Carrier proteins will be present on the cell wall of bacteria, which helps on the cell membrane particularly which helps in facilitating these ingredients into bacterial cell. Next is transport, active transports. So primary active transport and secondary active transport. If there is transport mechanism occurs through utilization of ATP as energy, through ATP driven transport occurs, then it is called as primary active transport. If that transportation occurs through electrochemical gradient, then it is called secondary active transport. Earlier it is diffusion, that is high concentration to low concentration. Here it is forceful. If it is energy mediated, then it is called, what is the main source of energy? ATP. You might have read it in biomolecules, right? So if ATP is the primary source of energy driven transport, then it is called primary active. If it is through electrochemical gradient, then it is called secondary active transport. These simple transports again classified into 4 types. If only one molecule is transferring, then it is called uniport. If two molecules like proton and molecule both are transferring, then it is called symports. But if it is transporting in different direction, one is through inside the bacterial cell, one is through outside the bacterial cell, then it is called antiports. Okay. If only one chemical is transferring, it is called uniport. If two chemicals are transferring in the same direction, it is called symport. If two molecules are transferring in opposite direction, it is called anti, as the name itself indicates, antiporters. Next is group translocation. This is the mechanism which explains about transporting and translocation of molecules at the same time. Okay. That is transporting the organism and it is incorporating the same molecule into the host. That is nothing but group translocation. It like this happens by attaching with the host protein. It will alter its size. Then it will transport that organism. Like hypnotism, we can say. Okay, so work will be done by hypnotizing the patient. So it will changes the structure of the host cell, then it will transfer the content into the bacterial cell. Next is iron. Iron is also a growth limiting micronutrient, which is helpful for the bacteria. So next property is temperature. So temperature also aids in growth and inactivity of bacteria. Because the temperature rises, enzyme activity declines. That is how during COVID period, so people are advised to take hot water and to stay hydrated and to stay in a high rise in temperature, because it will kills enzyme activity. That is enzymes present in uh, virus, or it will kills the bacteria. Okay, increased temperature will kills many bacteria. That's the reason why temperature also plays a crucial role in controlling its growth. Okay, each microbe has different temperature range. If minimum temperature is required, that is less than 24, that is called minimum growth temperature. If the growth is optimum, usually it is optimum at 37, that is our human body temperature only, growth will occur. Those are called optimum growth temperature. Maximum, if it is above 55, it is called maximum growth temperature. So depending on temperature, again these microorganisms are classified into 3 types. That is psychrophiles. That is, if they grow between 5 to 20 degree temperature, they are called psychrophiles. If they grow between 20 to 45 temperature, they are called mesophiles. If they grow beyond 45 to 50, they are called thermophiles. So depending on temperature, they are classified into psycho, meso, and thermophiles. And usually these bacterial cultures are stored at 3 to 5 degrees centigrade. Okay, that is through deep freezing. So this is the graph which shows if the organism able to grow between 0 to 20 psychrophiles, 20 to 40 mesophiles, 40 to 80 thermophiles, and 70 to 100 hyperthermophiles. So this is the examples for each. Mesophiles, E. coli is an example for mesophiles, Bacillus is an example for thermophiles, and Sulfolobus is example for hyperthermophiles. So how this temperature will affect the bacterial growth? We have given with an example. Say for example, leprosy bacteria. Usually this pathogen shows good growth at 35 degrees centigrade. But at 37 degrees centigrade, the growth was altered. Like less growth was seen. That is how temperature affects the growth of bacteria. And there is one more example also. Like Salmonella, Yersinia, Pseudomonas, they shows a good growth at 42 degrees centigrades. If they are grown at 37 degrees centigrade, they have shown good growth. If the temperature was increased to 42 degrees centigrade, the growth was altered. So this is one more frequently asked questions in your previously asked question papers. So explain how important to know about the temperature influences the pathogenicity of microorganisms. So you can mention about different types of microorganisms which falls under different temperature ranges. And two more examples which we have mentioned here, like leprae and E. coli, how they are can't able to withstand at higher temperatures. That examples you can quote over here. Moisture also plays an important role because 80% of protoplasm, that is bacterial membrane, is made up of moisture. If it loses moisture, it will get dehydrated. The uh, bacteria or virus get killed. So this low water availability will inhibits the growth of bacteria or viruses or anything, because it will reduce hydration, and thus it kills the bacteria. So that's why people say usually don't have stagnant water, uh, because it will encourage the growth of different bacteria. So that is all about the content. So I hope you have learned about brief about classification and structure of microorganisms. So this is about our Prakara. So why we are doing this webinar? To have a proper understanding on different contents on different subjects, we are introducing a concept called MLP, that is minimum learning program, which is only for 250 rupees only. It is valid for 21 days. As a trial pack, we are introducing to the people who are joining through this webinar. You can even explain to your friends also, who are staying in different states or anyone. So this package includes first semester and second semester. So for each semester, we are charging 250 rupees. So semester one has anatomy, physiology, sociology, psychology, which were dealt by our doctors like Dr. Sadiq sir, Dr. Adarsh sir, uh, Dr. Bovachia ma'am. So they are famous clinicians in different hospitals, like from KIMS or different institutions also. They are coming and we are making these videos. So we are collecting the syllabus from your universities and we are making student notes, key terms, and video lectures, so which your teacher was dealing. This is not a substitute for your teaching. It helps in better understanding about the content. So we have self-assessment quiz also. Once you have gone through the content also, to assess your content, we are providing quiz also. And now recently we are introducing this doubt solving webinars also. If you have any doubts or if you are not able to understand any topics, if you able to mail us, we will be dealing the same topic in the webinars. So this is about our content. So thank you.